Hello, my name is Katie Bond, and today I'm going to be talking about a paper that was released in 2022 in Nature that describes autonomous rhythmic activity in glioma networks that are then able to drive brain tumor growth. Now, this paper concerns a type of tumor called a diffuse glioma. Now, under this umbrella, this also includes glioblastomas. And these types of tumors mainly start in the spinal cord or the brain and are characterized by being extremely prone to malignancy and being very fast growing with high resistance to therapeutic interventions. And now, because of this, the prognosis for these tumors are pretty poor and are largely incurable, with the median survival time following diagnosis only being 10 months. Now, because of this, it's really important to understand in trying to find an intervention how these cells actually communicate and proliferate with one another over time. And before this paper came out, it had actually been discovered that these gliomas utilize tumor microtubes, which are these membrane tube protrusions that allow for individual glioma cells to communicate and connect. Now, the main mechanism by which these TMTs allow for communication between these cells is through gap junctions and intracellular calcium transients, which allow for communication with one another and the microenvironment, potentially contributing to tumor progression and actually development of the immunosuppressive tumor microenvironment. And in this paper, this was actually further confirmed with inhibition of cellular calcium entry and inhibition of these gap junctions that allow for the cell connectivity in vitro, resulting in this decreased cell proliferation of these glioma cells. In addition, while recording calcium in glioblastoma cells, they actually revealed this small fraction of the cell population that had these distinct periodic calcium oscillations, which they then referred to as periodic cells. In addition, through applications of graph theoretical concepts, which allowed them to map these complex tumor networks, it was observed that these periodic cells are highly interconnected to other cells in the network, acting as sort of hubs for cellular communication. In addition, when these periodic cells were removed from the network through ablation, this profoundly affected the actual integrity of the functional glioblastoma cell network, resulting in a reduced total calcium activity and actual size of the network that was able to be recorded, indicating the importance of these of the subset of cells, these periodic cells, and its relevance when it comes to tumor progression and network integrity. Now, following the identification of this subgroup and its potential in promoting cell proliferation through these calcium transients and oscillations, they then carried out an in vitro screen to identify potential inhibitors or agonists of these calcium oscillations in hopes of disrupting these periodic cells that act as hubs of communication for this tumor network. And these screens revealed two small molecules of interest, TRAM34 and Senecapoc, both of which are inhibitors of a calcium-activated potassium channel, which is called KCA 3.1. And they took these two inhibitors and then treated glioblastoma cells in vitro and found that they observed both a decrease in the prevalence and the calcium activity of periodic cells, along with a decreased cell proliferation of these glioma cells in vitro, along with a reduced expression of previously cancer-associated pathways, including NF-kappa B and MAPK. Now, after seeing these promising results in vitro with decreased calcium activity of these periodic cells and decreased glioma proliferation, they then wanted to move to in vivo. So they took mice that had been implanted with glioblastomas and then either subjected them to no treatment, treatment with the Senecapoc inhibitor, and then lastly, just a knockout of this KCA 3.1 potassium channel. and observed tumor growth over a period of 72 days. And what they saw was pretty striking. With treatment with the Seneca-POC KCA 3.1 inhibitor or just a knockout, resulting in a severely decreased tumor size and slower progression. Now, along with this, beyond just looking at the tumor size over time, they also looked at survival time, so prognosis essentially, of these mice, and found that those that were treated with Seneca-POC or the knockout experienced a increased survival time compared to the control, which really highlights KCA 3.1 being a potential therapeutic target for these cancers that were previously incurable. So as I previously mentioned, through this paper, I think the primary takeaway is KCA 3.1 is really highlighted as a potential therapeutic target for these gliomas that were previously accepted as incurable in humans. The panel that they end on, which I think is really interesting and brings this message home, is that they look back into the cancer database and look at expression of KCA 3.1 in patients, actual human patients, and find that those who have a higher expression of KCA 3.1 have a decreased survival time compared to those with a lower expression. So this is just supporting that for future study, this therapeutic target that was seen to be promising in mice could be extended to humans. Now, of course, that's a huge jump. So between using this in mice to using this on humans, there's a lot of intermediary steps that would need to be taken. Firstly, as stated in the paper, inhibition of KCA 3.1 also resulted in decreased expression of NF-kappa B and MAPK pathways. And although they are cancer associated, they also have normal functions in the cell, such as NF-kappa B having roles in pathogen defense and in typical immune functioning. So those possible off-target effects of KCA 3.1 inhibition would have to be investigated. In addition, before moving to a human model, utilization of some biomimetic 3D tumor models, such as organoids and tumor on a chip, could be a good way of testing if this therapeutic target and these inhibitors also use in a more human like model beyond just the mouse. And here are my references. Thank you.